Breakthrough: primitive stem cells shown to regenerate blood vessels in the eye. I'm Steve Blair. Welcome to Breakthroughs. Johns Hopkins Medicine scientists say they have successfully turned back the biological hands of time, coaxing adult human cells in the laboratory to revert to a primitive state and unlocking their potential to replace and repair damage to blood vessels in the retina caused by diabetes. These findings from this experimental study, they say, advance regenerative medicine techniques aimed at reversing the course of diabetic retinopathy and other blinding eye diseases. Our study results bring us a step closer to using stem cells more widely in regenerative medicine without the historic problems our field has encountered in getting such cells to differentiate and avoid becoming cancerous, said Linus Zambidis. Results of experiments using human cells in mice were published online in Nature Communications. According to the National Eye Institute, diabetic retinopathy is a leading cause of blindness in U.S. adults. By 2050, researchers estimate that some 14.6 million Americans will have the condition, which results in abnormal blood vessel growth in the retina or light is processed into vision. For the study, scientists began their experiments with a fibroblast, a connective tissue cell taken from a person with type 1 diabetes. Reprogrammed fibroblasts function as stem cells with the potential to give rise to all tissues in the body, including blood vessels. The Johns Hopkins team reprogrammed the fibroblast stem cells to revert to a state that is even more primitive than that of conventional human-induced pluripotent stem cells, more like the state of embryonic cells about 6 days after fertilization. This is when cells are the most naive and more capable of developing into a specialized type of cell with a much higher efficiency than conventional human-induced pluripotent stem cells. To do this, the scientists bathed the cells in a cocktail of nutrients and chemicals. What should go into the cocktail to build a better naive stem cell has been the subject of debate for over a decade. Zambidis' team used a cocktail mixture of two drugs that other scientists previously used to reprogram stem cells. GSK3 beta inhibitor CHR99021, which blocks carbohydrate storage in cells, and MEK inhibitor PDO325901, an experimental anti-cancer drug that can block cancer cell growth. The team had also looked at the potential of a third drug, a PARP inhibitor, a popular anti-cancer drug used to treat a variety of cancers, including those of ovaries and breast. To the researchers' surprise, Zambidis said the trifecta of MEK, GSK3 beta, and PARP inhibitors work to wind back the cell's biological clock. He calls the cocktail 3I, named for the three inhibitors. Zambidis' team had first reported experiments using the three-drug cocktail in 2016. The scientists also found that the stem cells reprogrammed with the 3I cocktail did not have abnormal changes in factors that could alter core DNA, called epigenetics, that typically plaque over lab-made versions of naive stem cells. Finally, the research team injected cells called vascular progenitors, which were made from the naive stem cells and are capable of making new blood vessels, into the eyes of mice bred to have a form of diabetic retinopathy that results from blood vessels closing off in the retina. They found that the naive vascular progenitors migrated into the retina's innermost tissue that encircles the eye, with higher efficiencies that had been reported with vascular cells made from conventional stem cell approaches. The naive vascular cells took root there, and most survived in the retina during the duration of the 4-week study. Interestingly, the 3I naive programming cocktail appeared to erase disease-associated epigenetics in the donor cells and brought them back to a healthy, pristine, non-diabetic stem cell state. For comparison, the team reprogrammed diabetic fibroblasts to non-naive stem cells using standard methods, and the resulting vascular progenitor cells failed to migrate as deeply into the retina or survive the length of the study. Ha ha! That's cool. I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.